Victory is always possible for the person who refuses to stop fighting. And in agreement to this, it is my pleasure to welcome you all in today's event of MSI, an inspirational story of grit and determination by Dr. Shubham Goel. I'm sure that students from all over the world will certainly be benefited by this talk. And now I request our President, MSI Delhi Unit, Dr. A. Archana ma'am, to open this session. Over to you, ma'am. Thank you.
Thank you, Dr. Shweta. So I'm very happy and thrilled, very excited, and a very good evening to Professor Deshmukh sir and Jyotsna ma'am, who have graced this event, and lots of salutes to both of you. I welcome Dr. Shubham Goel, who came to us as a small child, a boy of 17 years old, and he has evolved and emerged so well. It's a treat to our eyes. I welcome all my esteemed colleagues from various colleges of University of Delhi and other places. Others are still joining. They confirmed to me that they will join. Yeah, good evening. So lots of wishes are coming in front of you. So I take this opportunity to invite all of you to enjoy the talk given by Dr. Shubham Goel, who has become a world-class scientist only due to his vision, sustaining hard work, and honest attitude, never giving up attitude. So he's here among us, and I sincerely hope that our upcoming new generations benefit a lot from an esteemed, accomplished person like Dr. Shubham Goel. We know that everybody has been fighting the dark era of COVID-19. So let us hope, pray, believe, and make efforts that not only the sunrise is now happening soon with the vaccines coming up and some positive changes in the profiles of infection and recovery, but also not only sunshine, but also a rainbow of hopes, accomplishments. And for that I thought of Dr. Shubham Goel. Who could be better than him? Because he's always willing to speak and to motivate. He has been a good mentor to many students on my request, and this is all a teacher wants. We don't want anything for us. We want the accomplished people to inspire the developing students. So I now invite Ms. Nupur Tyagi to introduce the speaker to the august audience. And Nupur Tyagi is his super junior, and she's also from our college. She's doing her MSc in Microbiology from University of Delhi South Campus. And over to Nupur to give the introduction, to introduce the speaker and his accomplishments to the audience.
Thank you, ma'am. Very warm greetings to one and all present. It's my proud privilege to introduce our today's esteemed speaker, Dr. Shubham Goel, who has made big in his life and is super senior of mine from Swami Shraddhanand College, University of Delhi. He has been a mentor to me during the tough times. He's a man of virtue and simplicity. I feel elated to introduce him to everyone today. We are wholeheartedly grateful to the founder of MSI, Dr. Arvind Deshmukh, for giving us an ample amount of opportunities. We couldn't have done any of this without you, sir. A heartfelt thanks to Dr. Archana ma'am, President of MSI Delhi Unit, for always being an exceptional source of inspiration and motivation to thousands of students. I thank her for giving me this exclusive opportunity today to introduce my role model. Our distinguished guest, Dr. Shubham Goel, is a visiting scientist at National Institute of Health, NIH, USA for last 10 years, and is working on rare genetic diseases of immune system to explore pathophysiology and molecular mechanism of the disease. His PhD is from PGI-MER Chandigarh, and there too he was working on human diseases which affect primarily the immune system. To name a few, he has been working on STAT3, CARD9 and TCF3 genes, and deficiency of all these genes make a person susceptible for viral, fungal and bacterial infections. He has been working on primary immunodeficiency diseases, PIDs, for last 10 years and has standardized various diagnostic assays and as well as molecular mechanisms in patients with PIDs using cutting-edge high-throughput techniques. And techniques including flow cytometry, next-generation sequencing, and single-cell RNA sequencing. He has also shown his contribution to microbiology community by publishing many research papers in peer-reviewed international and national journals. Shubham sir's academic graph has been really uphill and it is dream graph for any one of us. We requested Shubham sir to share his inspirational journey with the future aspirants. We hope you will all enjoy the talk and be benefited from it. Since many of us are sir's juniors, we would love to greet you, sir, with a bouquet of lovely flowers. Everyone, please pin Bharmjeet's screen.
Thank you very much. Thank you, ma'am. Thank you, Dr. Shweta. Thank you, ma'am. Thank you, Nipur, for this such a nice introduction. I never expected to have such, and this is for the first time that having this kind of formal introduction and giving the detailed information. Well, I'm really overwhelmed with this nice introduction. Well, without any delay, so first of all, I would like to thank to the President of the Microbiology Society, Professor Deshmukh, Archana ma'am, and of course the coordinator, Dr. Abhishek, who invited me and gave me the opportunity to share my experience, my journey, how I've done it. But before that, I would like to say it's not that I'm the unique one, actually. There are many actually who has faced the same situation and have achieved their goal and the targets. Though the way could be different, they have a different strategy, but ultimately to get their goal or the achieve, they have some common features or there are few features which remain constant. And out of those, all I would say, these are the grit and determination. So first, I would say that just a little bit about what the grit is. The grit is about perseverance, determination, passion, going the distance despite the setbacks and focusing on winning the marathon, not the spirit. So these are the features which I believe that if you have all these, no one's going to stop in achieving your goals, your targets. And about all these features, what I'm showing here is going to talk about with my subsequent, or it will be covered when I start my journey by itself. So if just look at my journey, so I started from here, you see there is a small village, Pulla. I just went from here to Swami Shraddhanand College for my graduation, and from there, after completing my graduation, I went to do PGMR Chandigarh to do my master's, but this was not the end of the story. After master, either you go for PhD or you look for other options. But luckily, in my case, I got the PhD in the same institution, and then the story begins from one country to another country, and then I left India to have a more exposure of the advanced research in the United States. That's how I entered. So it looks like I'm traveling from one place to another place, but this traveling is not just for the fun, just in search of science, in search of truth. So don't think that it's just a Google map. It's just, journey to getting something, to have some achievement. So this is, so we'll start now from where I started. So this is the Google map, basically the Google satellite image of my village from where I belong. And I would like to say that at the age where you are right now, so especially the young students, like those are in the BSc, when I was there, I used to think, oh, I think I don't have any goals or I don't have any targets. So, but until I entered into my PhD, then I realized, oh, I was pretty much clear what I want to do. And I would explain why I'm saying this. Because when I was in 12th, so before that, actually, I used to start, I had that I want to study about the life. I wanted to know what the life is actually, and to carry out that research or to answer that the question, I used to think how I'll do that. And at the same time, when I was doing my 12th and I was about to finish, there was 2 courses was coming up in my nearby area. Those were like biotechnology and the microbiology, and they were booming up like this. Oh, these are the 2 new courses, and there's a very big hype on those courses. But my thought was to think about how the life is going on and what the life is actually. And with that mind, because the microorganisms are the one which like with the unicellular one, and they can perform all the body function as all the multicellular organisms like the human, like other animal. So with that thought in my mind that, oh, microorganisms have those functions, so why don't we study those things? So with that mind, I thought, okay, let's do the microbiology. And for that, I started looking for the microbiology courses where I can do. And I get to know, oh, Delhi University offers such courses. So I just moved to the Delhi University. And there, so what I'm saying here, what your outcome is actually, so you should know what you're headed and why you are, and you're big. So actually at that time, maybe I was not aware, but I was clear. So at the end, I realized that, yeah, I was aware that what I want to do. So that time I was clear that I will do the microbiology just for the sake of my knowledge, just for the sake what I have designed in my mind. Well, then when I reached to the Delhi, so I basically filled the form, and then that is how actually I landed to the Delhi University in the Department of Microbiology there in Swami Shraddhanand College. But getting admission in Delhi University was not an easy job for me, at least, because I faced a lot of issues, and I will share those things, like what are those things there. And I would say getting not only the admission in the BSc, basically throughout my master's, PhD, postdoc, even in every steps, I have been like supported, and there's so much of contribution by many people, and those includes like parents, teachers, friends, classmates, seniors, juniors, and the staffs. So without them, probably I would say it is impossible to carry out your research plan that you have thought. So which I will explain one by one, like how they have played an important role into me. So first of all, the parents basically who believed in me, who always supported me. In fact, they never stopped me what I wanted to do. They never thought like you just do that thing. They just said whatever you want, you do it. And with their help, I was able to come here and stand up here. Parents is the first role for me, at least, definitely. And then here comes the role of the teacher. So, well, I will explain more about the teacher. But before that, what I was talking, that it was not easy to get the admission in the Delhi University. You must be aware that if what used to be happen in Delhi University, that based on the three scores of the subjects, the physics, chemistry, and biology, they used to set up a rank, the cutoff, and based on that cutoff, if you are into that fit, then you can go to the admission. So there at my time, there were like 4 colleges, and out of those 4, the 2 were only for the co-ed. So that means the rest of the 2 were basically for girls only. So I was just eligible for the 2 college, and even those 2 colleges, they were just like 26 per college. So that means just 40 college I have to compete for that. And luckily I filled the form, and I was able to fit into the 4th cutoff of the Swami Shraddhanand College. And if I remember correctly, I was the second last to get that chance, and I was able to fit. Okay, I went to the college to get I was eligible there according to their cutoff. And I was called by the principal, and there was a guy nearby who was sitting nearby, and then he started saying, "Oh, you belong to UP. So I think we don't believe your degree. It must be fake. It could be you must have buy from somewhere." Then I said, "No, sir. This is my own original degree, and I have done my work. I have done hard work." So he said, "Okay, but will you be able to sustain here because you don't know English much?" Then I said, "I know, sir, but I will try." Then he said, "But you might be fail." Then I said, "It's okay, sir. I will give a second attempt." Then he said, "But we don't give a second attempt. If you are failed, then you have to go back." Then I said, "It's okay, sir. If it happens, I'll try my best. If it happens, definitely I will never come back again." And my dad was just standing with me, and he was listening the same conversation. And at the same, and once he done, then principal questioned me just few questions related to biology in English, and I answered him in Hindi. Then he said, "Okay, we are ready to give you admission. So what you can do, what we need actually, you just go to the head of the department. If she agree, or I don't know at that time who was the actually head of the department of microbiology, they said that if he or she is agree and they put the sign on your form, then you can get the admission." So with that, I just took my form. I went to the ma'am office, but as I told you, I was not aware who was the HOD. We were not allowed to go inside. The peon took my file. He went inside, he bring it back, and it was signed. So we were too much lucky, like my dad and me was too much happy that, oh, finally the ma'am has signed my form. And we were really actually thankful to ma'am that without any hesitation, without my background, she considered my application and she signed that form. But now the fear was coming to my dad. Shubham, they are going to fail you. If you stay here, probably there's a chance that you won't succeed. So let's go back and don't take the admission here. So my dad took me back, and I thought, okay, let's take the admission there nearby in my state. I went there with my disheartened heart because I wanted to get admission in DU only, in the microbiology here. So I went to my state. I went for the other admission. Then my dad called me, and I was in the session over there. They were talking about the microbiology and all in the UP. So my dad called me. Do you want to join in UP or you want to go back to DU, Delhi University? Then I said, yes, I want to go there. He said, then go. And with their immediate yes, I just left that hall and I just ran away. I took the bus for Delhi and then came to the college, and was like, when I reached, and that was the last day, because once the cutoff is, advertised, you are given like 3 or 4 days to get admission in those period. Otherwise, you are gone. And luckily, that was not the luckily. Basically, it was the last day when my dad said, "Okay, yes, now you can go." And I went. And when I reached to the college, it was almost, I was like, something like that. And then the accountant was, supposed to close his account where I need to deposit my fees. I reached there. There was some union leader. Basically, the college leader used to be there. They asked me like, what happened? Then I said I want admission. Then they requested to the cashier that please accept his admission fees and give him an admission. And I got the admission. So that was the whole story about that, how I get the admission there in the DU. Like, I just went back and then came back. Though my destiny maybe wanted me to come back here, but thing is that I have make up in my mind that what I want to do and where I want to go. So probably it was determined in me that where should I go. And so with those like back and forth forces, I finally made it and I got the admission there. But getting an admission is one thing, right? And doing the and keep continuing is the another thing. The major problem with me was that I was from the Hindi background. I never had experience of, like, we did the English course, but was not up to that mark. And I didn't know like how to speak it. So then when I was in the first year, you won't believe it, that everything that just goes, from my head. And I used to think, oh, what's happening to me? And then the teachers come here, where the Archana ma'am and my other teachers in the class, specifically, like, they used to translate for me. Whatever the statement they say in English in the teaching statement, they used to translate for me so that I can understand. That was a big help for me. And I remember one thing, the one incident with the Archana ma'am, that when I was in third year, everyone was given a topic to present, and I was given a topic of DNA fingerprinting. So I was called. I went there. I knew like what to speak. What is the idea? What's the DNA fingerprinting? But I don't know how to put into English word. So I just stood up there and I start saying few things like, in breaking English word like this and so. So I didn't know like what to call police in English, like the cop you can call, and then corrected me. Yeah, that you can call. I didn't know what is the site of like where the things has happened. So you can call it the crime scene. So I still remember those things, that how I completed my presentation with the help of ma'am, that she helping me in completing those sentences and putting my words. So yeah, that's really nice to me. And that's not only that ma'am has helped me, it's just everybody in the unit, whether it is like sir or ma'am, the other teachers there. But that's the teacher's role. The other important roles is of the friends. What I would say, he's one of my best friend, Rahul Madan, and he's the topper of the GU of the microbiology there. And he has helped me in reading everything. He literally used to translate every single sentence for me, and we used to study together. And today, what I'm saying, what I am in terms of my personality is just because of him. He helped me a lot. He transformed me like anything. So I'm not saying that he's the only friend who's helped me. There are many friends in each and every step of my life. Now, what the important thing is the classmate. What I always believe that it is about the learning. It is about the observation that you can learn. And for me also, it was working, because in my classmate, everyone has a unique quality, the unique information they had it. And and few of them I would like to mention. The Shrika Bhardwaj, she used to make a presentation very beautiful, and I learned from her by watching her, like how she did it. Then the Shubda, she used to, speak very fluently in English, and her presentation was like excellent and absolutely perfect. And that's how I used to learn. I used to catch her words, and this is how actually I get the few things from my different classmates, and I was trying to growing up and learning more and trying to make myself better. And of course, the seniors also has an important role playing. When you have any question, your query, then they help you, and that's is how. And here you can see the show accessories here with their students. And though we used to bunk a lot of class with, but still, we enjoyed his classes a lot. Then the juniors again important play role, because the juniors are the one who ask you question, and then that is how you actually expand your brain and the mind. And you must remember, like, whatever you read today, you might not be that much understandable, but once you read or go to the next step, then you understand much better, or it get much clear when someone asks you that question. The similar thing happened to me. I was not that clear in the first year about those topics. The second or third year, used to ask me any question, then I used to read more and dig into more information, and that's how I used to clarify my doubts, and that's how I expanded myself. So that's how the juniors are also important for you, for your growth. And the most important thing, which is actually I learned only from this college, though many things, but this is one of the important things, is the junior-senior relationship. This is important. Why? Because I just followed Anupam sir. This is one of my senior, and he's always helped me. Not the other seniors, like, they help me in telling you like where you can apply for the grant, for master program, for PhD, or for other or whatever you want to do. So they help you in guiding in every way. So the way I follow Anupam sir, the way he's following actually my juniors and Nitish. So that's how we made a junior-senior relationship, and which always help you making in your career best and better. All right, so this was all about getting into graduation. So somehow I was able to do and clear and pass my graduation with the support of my all teachers, my friends, my colleagues. But the next move was to go for masters. And for that master, I just set up some goals. So I would say these as a SMART goals, and those are like specific, measured, achievable, relevant, time. So what all those mean is that, like, you are going for the master program, so you have to think where you want to go, and what kind of content you have to study, because in each exam they have a different pattern, they have a different prospect. Different information they ask. So you don't need to read all. You just be very specified that what topic you need to read for a particular exam. Just go for that. You have to be major. And then what information is relevant for those exams, entrance exam, you have to do, and that has to be in a timely manner. And for me, what I believe, the time efficiency is the one thing. Like, the people who worked a lot, like day and night, I don't think so they are efficient. The efficient are the one who can finish their work in a timely manner. So that I would call it the efficiency. So with those basically efforts, I made it in PGI Chandigarh. I started my medical biotechnology there, and there were 2 years went well smooth, but still I was struggling with my English. I was still learning with my colleagues there who used to present their seminar and GC. I used to sit with them and discuss with them, and then my teachers helped me. But anyhow, I was at least much better than before when I was in the graduation. So this happened just because I just kept going and I never gave up. Well, now after the masters, so there's one thing which I would like to mention. It is always better if you know what you want to do before, or at whatever stage you want to do. Because I remember when I entered into graduation, there were like around 20 people, and from there just like 16 people there. And then at the end, when I just left in the graduation, there were just 8 people there. So everybody left. It's better, like, they recognize what they want to, and they moved it. And I remember now, out of those 8 people, those has completed graduation and then masters, only the 5 people are still doing the research. So I'm not saying that those who left are not doing good. They are also doing good, but it's just that they were not actually not aware what they want to do. Now they are doing much better than the science, and that's a good thing. So, and I was pretty much clear that I want to do, after master, that I want to do PhD. But before I tell you about like the PhD and how I find everything, so there's few things which is important for the young generation who is gonna pass the master exam and wanna go the research. There's important thing to know about a PhD guide and the research question, how to choose and pursue. Why I'm telling, because the PhD is not a year actually, it is of a 5 year or 6 year, and under whom you are going to join. You have to be happy, you have to be stress-free. Once, if you are under like a guide, then your life will be hell. So it's important to decide a guide. And for me, a PhD guide should always be a good human being. At least for me, I always search for who has a good human being, who has the ability to form a good and trackable question with the good experiments, good publication records, which makes you independent, confident over time, who actually helps you or enhance your training by involving you or by sending you in the summer school and the conferences, who makes you create the happy and interactive environment in the lab, and helps you in the good talk with the papers. He helps you in the writing the grant papers and the review paper. And of course, the very important is the one, a good guide would always be, I would consider, who will always support you, even if you leave your lab, he recommend you as a good student. So that's about the PhD guide. And what should be your research question when you are in the PhD? It should be like, first, always find a good mentor, a good PhD mentor, and then look for what kind of lab work is going on. Because what I believe, again, the PhD is about learning. There are people like who has their own interest, like they wanted to do some on particular topic. There's nothing bad, they should go. But I would say preferably first find a good human being under whom you're going to join. Because if you join under him, definitely you are going to gain expertise and which you can expand later on. There's nothing wrong. The point is always find a good guide and look what kind of work they are doing in the lab. And according to that, read those papers and think about what has been done, what has not been done, and then hypothesize what your hypothesis which has not been done. And then look for lab resources so that you know whether these things are doable in your settings or not. And then accordingly, you design your experiment which are doable in your setting. I would recommend which one should not do when they are choosing is that who works on one specific topic. Again, I mentioned you before that there's nothing wrong that if you have an interest, like for example in cancer, and you find a guy, but he's not a good human being, then there's no use of doing a cancer research until you are happy or you are just, your life is hell. So important is to find a good guide. You have to cast a broad net so that you can fit into a good guide. There you can learn many things, techniques of what they are doing and do better in that field. That's much better to do doing on a specific topic. The second thing is that never try that a guide like you want to undergo who has a Nobel Prize. It's not necessary that all Nobel Prize winners have a good attitude. And the third is that never decide a mentor just because he has the same gender or he has the opposite gender. Never decide based on that. So this is what all about the PhD mentor and the research question. And for me, I was lucky enough that I find a guide who has all these qualities. Although I had an opportunity to undergo for PhD under a top, I would say, basically a topmost faculty in the microbiology, but still I left him and I chose a guide under whom actually I get the freedom of doing my work and as well as my personal life, which he always provided me. And under him, I just get every kind of expertise. And at the time when I joined him, I knew him actually, and even what he was doing, but I joined just because I find him to be a very good guy. And once, when I did my PhD and I under him like for 5 years, I learned the techniques or the topics which has not been done in India before. And I would say proudly that I am the first student in the PhD who has done primary immunodeficiency in India and has set up very like a lot of diagnostic assays, and the people are following. So that was a great achievement for me. Though this thing which I have never decided that I want to do this, but what I'm saying that I'm just going with the flow, and I got the things in pretty good manner. Well, so that was the okay. There's a one thing which I wanted to tell some of the students, those who want to do PhD in the United States. I just mentioned few of the fellowships. They can read or they can ask me. I can send them separately. So that's how my PhD went very well. But do you think the story was going smooth? No, that was not like that. I had a failure again. A failure was that I was trying for the postdoc position. I was not getting it. I keep on applying, but there's nothing. There was no response coming up from there. And to applying for postdoc, you don't need to create a very long application. It's just that you type to a PI where you want to join, just to write why you are interested, what you want to do, and what's your vision. And I believe that I was doing something wrong. That's how I'm not getting any response. So probably what I'm trying to say, the failures makes you actually think more and and they allow you that you should respond in the other way. So that means actually there is no failure. The failure gives you a lesson. What I did, so what you can do? You can have a different choices. You can have alternative approaches, new perspective option, and you can think what else I can do. So again, with the help of my seniors, I asked them, I put my concern that this is happening, I'm applying and I'm not getting it. They said, I think you're doing this thing wrong, let's try this. And that's how I tried an alternate approach to applying that postdoc position. And I did it. And finally, I landed at the NIH, National Institute of Health. It's a prestigious institute, and this used to be my dream institute. Why? Reason being, I was working in the PID, and most of the people who are working on a PID in the same institute, and I wanted to work with those whose paper I used to read it. And that's again, see, I did nothing. I was just doing what I want to do, but I get my destiny. It's just that I just kept going. And this is how then my dream came true. I worked in the lab, the high throughput lab and the excellent lab, and that's my past lab where I used to work. And just recently, 2 months back, I moved to the new lab, and this is how you can see that how we used to have, and you also have the same thing, that lab meetings in the COVID era. So there in the NIH, I started learning very new thing, and I would recommend everyone that at least one should come to have more exposure of the research expertise, because it's entirely different than the India. Once you are here, then definitely you will know the difference, and you know that how the research is going on there, and it's always better if you learn. So with that, now I would like to mention some of my research work which I have done, and that's the primary immunodeficiency diseases. So PIDs are actually, and this is my area of expertise on which I'm working since last 10 years. So I would explain what the PID is. So PIDs are the defects in the gene or the mutation in the genes of the immune system, which basically either alter the function of these immune cells or they block the development of these immune cells, right? So they might have like, for example, they might not be a B cells or might not be a T cells. So that's the one thing where there's no development. Or the other way, that the function of any of the cells is defective due to the mutation in those genes. And and I believe that you must have, one of the common example is the severe combined immunodeficiency diseases. So what you can expect, so when you have a defect in the immune system, that definitely you are going to have a more and more infection, because these are the cells which helps you or which protects you against the infection, right? Okay, and now I can give you some light on the recent pandemic which is going on, which is the COVID-19. You can think what is which might be affected more with this virus. So probably the answer would be definitely the patient who has the weak immune system, or the patient who has the PID. So PIDs are the one who has the defect in their system by birth, right? So this is how important to understand the primary deficiency diseases, which explain the the effect of the immune system under their respective mutations. So for example, if the patient has a mutation in the, so for example, in the B cell gene, so definitely they won't have any B cells. And then you find the phenotype in the patient of the B cells. And then, but to prove that that mutation is really is the causing of the disease, you need to prove in vitro, and that we do it and we prove it. Yeah, that's the real cause of the disease. And that is how I would do. I would like to give some lights on my research work, which has been done. So these are like the card9 deficiency, TCF3 deficiency, and the STAT3 deficiency. These all are immune genes and they work in the different cells, and they perform different function and how they are playing an important. So what? Again, the one thing I would like to emphasize here, just try to find a work, try to work on the research which has an application. The application, either it is a basic, as a clinical, or a translation application, because and those applications basically actually involve directly or indirectly in the human mankind. In my case, I'm lucky that I get chance to do work on PID from my PhD, and from there I just decided that I will keep on working in the same field and I would explore the things. And this is as you can see, these are actually the nature's experiment where the nature had the mutation in the human itself, and then you can see the phenotype. Oh, that's happening when you don't have the gene. There is no T cell, there is no B cell, and then you can correlate easily. So that's the beauty of the nature that they made it. Although there's a sad point that if it is in the human, in the patient, that's their patient, that they get sick. But this is how we learn. Anyway, so I'll discuss one by one about the card9. So card9 is basically a caspase recruitment domain, whose deficiency basically makes you susceptible for the fungal pathogen, and especially which fungal is the Candida albicans. So if you have a card9 deficiency, so the person, they are going to get, you can see such kind of fungal infection on the skin lesions, any body, and especially they have in the brain also they can go. And in my case, I had a mutation in this card. I had one family. In the one family, I find this mutation. And the important point, the part of this mutation is that at the one point, it is making susceptible that family to the fungal infection. But on the other hand, the same gene is making them protective for the inflammatory bowel disease. So again, that's, you see the uniqueness of the gene function. The same mutation, but performing a different function in different aspects. So what I have done in these patients, so I investigated the behavior of the card9 mutations in patients with Candida infection. But importantly, the one thing which I have done in this study, so this gene basically was supposed to be expressed in the monocyte in the macrophages. But for the first time in my study myself, I have shown that this gene is not restricted to the myeloid. They can be expressed in the T and the B cell. So that's how I showed that and I showed in the human sample as well as in the mice sample, and clearly defined that this gene is in there. So probably this gene might have a role in the T and B cell biology. So that's one thing. And the other thing, I explained that how this card9 is in plasmablasts are the cells who basically produce the antibody, and the antibodies are important to provide a protection against the infection. That's the one gene I was talking about. The other gene which I have been working is the TCF3. The TCF3 factor, and which has an already well-known function. When you have a mutation, you don't have any B cell. So you can assume when you don't have any B cells, so that means you won't having any antibody production. So probably such kind of patients will be suffering from the bacterial infection, because the antibodies are important to controlling the bacterial infection. So that's what we have seen in this patient. But that's not my duty. Not the duty, basically, not to explore which is already known. My duty was to know which is not there. And that thing was the role of this gene in the T cell development. So that was, I was trying to explore the one thing. But the other thing is that I was trying to explore the gene dosage mechanism of the TCF3 gene. The reason, this is actually homozygous recessive one, but I have seen some of the patient who has the heterozygous mutation of the same gene, but they were presenting some kind of immunological or the clinical features. So I was trying how this gene has a gene dosage mechanism and have a role in the clinical and immunological penetrance. That's one thing about this. The third gene which I have done in my PhD is the STAT3 gene, and which is basically cause hyper IgE syndrome. And the STAT3 gene basically is the gene where when you have a deficiency of this, so you have a specifically infection of the staph aureus, staphylococcus aureus, and the candida infection. So again, and it happened because the STAT3 gene is involved in the development of the TS17 pathway. So what I would say, the immunological defect, so I said the STAT3 gene is involved in the TS17 development, and that's how they get the infection susceptibility. So that part, basically the immunological defect was already known. What was not known, that how this gene basically causes the skeletal and dental defect. In my study, I have shown this, that when you have a deficiency or the mutation in the STAT3 pathway, it's lead to the inhibition, or there is no secretion of the osteopontin gene, and this osteopontin gene basically is responsible for developing all these phenotypes which we see in our hyper IgE syndrome patient. So basically, with these PID patient, I'm still learning. I'm still working on the PID patients and the PID mice, where I'm learning how a defect in a single gene actually lead to what kind of phenotype and what kind of a functional defect in the new gene, which is still very encouraging for me and very applicable to the human patient, because when you identified where the defect is, you can directly target those molecules. And one of the example I would give you with the deficiency of the MACT1. So MACT1 is there, where the magnesium is actually deficiency there. So in that patient, if you provide the supplement of the magnesium, you just reverse the phenotype. So that's the beauty of these experiments, that once you know the molecular mechanism, you can look for the therapeutic approach. Well, with these, there are a lot of information, but still, if you think that I have left few things, or there's thing that you want to know more about, you can ask, and you are feel free. But there are few points which I would mention professionally we should have, and personally. So the professionally, always work on your weakness. If you think that you have that weakness, definitely you should work on it. Just keep learning, stay focused, have fun in life, never miss that part. Be motivated, stay positive, and have patience. Definitely the good results will come back to you if you have it. So personal, always be thankful, be sincere, be trustworthy, have fun in life, never give up, and go with flow. That was my funda actually of my life. Just go with flow where it is taking to you, but do with dedication wherever you go. There's one thing which I would like to say is that there's one person without whom actually I can do nothing, or who supported me even after my marriage, and now you can assume who it could be. They are actually my wife, my kids, and that's how, other than doing the science, I'm doing my family management, so that's how I became a family man. Well, at the end, so a big thank to Professor Deshmukh, the president, and my ma'am Archana, and the coordinator Abhishek, Saini. Thank you very much, and please feel free to ask anything if you have any question. And I hope that I up to your expectations. Thank you very much.
Thank you so much. It was a very scintillating and encouraging talk. And we will go for the moderation part, the questions, the queries, and whatever things these students are likely to face in life. But first, I want to confess my mistake, very big mistake, and I want to rectify that. And on one side, I'm really proud of Shubham and very, very overwhelmed by his progress and his attitude. But more important than this is that I'm really sorry. I should have asked. It was written in front of me, but I forgot to look at it. So before anything else, I would request, with a lot of respect, Professor Deshmukh sir, to say something about today's talk, and if possible, ma'am also.
Thank you, madam. And at the outset, let me tell you,
yes,
it is not your mistake. You were deeply involved in your student.
Yes,
And listening your student. It was a very happiest day for you.
yes, true.
Also for Shubham.
Yes,
Sir.
That relationship should be there throughout the world among the teacher and student. I appreciate today he has given, he has taken full account of his journey, academic journey, personal journey, which is a definitely inspiration to all students. Again, I can now firmly tell to all students, every one of you can become a Shubham. Shubham is a symbol. I'm not telling the changing the name. I'm talking that like Shubham, everyone can achieve the things and he or she can achieve more than Shubham. It is possible. Only because he has taken the account. He has told positivity should be there. He told patience must be there. And many things he has told to the young generation. So really, Archana ma'am, I am really again and again I am telling, reiterating, I am thankful to you because you have given a good platform to Shubham, as well as you have given good platform to many students which are joining today, listening today. And we'll continue. Means again, Archana ma'am, I will tell that you are my pillar. Whatsoever you are doing, people says that Dr. Deshmukh is doing. It is not a Dr. Deshmukh. It is the many associate like you in India. And we should serve not only India, people in India, but we should go beyond India, humanity. So I congratulate Shubham, I congratulate Archana ma'am and her team for this great success today. And I will request to all students, ask the question in chat box and have a discussion, take advantage of the today Google Meet. Thank you.
So thank you so much. And I have learned today from you that despite the fact that I made a mistake, you were so cool and you in fact praised me. And sir, really, I have no words. And you are also like, we look up to you, sir, with folded hands, sir. Thank you. Thank you. And ma'am, would you like to say a few words to bless?
I have listened the journey of Dr. Shubham. Very well done by Dr. Shubham. And only I want to give best wishes to his future journey. Inspirational to young generation. And definitely the young ones have inspired from his speech. And likewise, you arrange the talks of many students who have successfully done their journey.
Thank you. And he's still young and he wants to now come back to his country and serve his motherland now. Yes,
I
sir.
object for one sentence.
Yes.
That he is very young. I am telling I am also very young.
Yes, yes. So
Correct?
am I.
Yes. Yes, yes. Because physical youngness and mental youngness.
Yeah, it's the attitude. Yeah. So
Correct.
you
It is an attitude.
yes, you both and people like us, we will never grow old because age is only a number.
Correct. So I would like to tell Dr. Swami, I am also equally young like you.
Yes, sir, you will be.
Young and dynamic. Dynamic is dynamic.
Yeah.
Yes, you will be.
Sir, may I request my senior colleague, Dr. Sunil Srivastava, who has even mentored me at a stage. So,
Very
Dr.
good.
Sunil Srivastava,
Yeah.
Shubham is your student as well. Please
Yes.
say a few words.
Oh, sure. I would be very much delighted to do that. Yeah, Shubham has a, I mean, when I saw him first, I saw a lot of innocence in him, which I didn't see in many of the students who were coming to DU. So there was a childlike innocence and a childlike curiosity. to know things, right? In my class, he would just look at me, and I mean, I would look into his eyes and I could understand that he wants to ask something, or he's very inquisitive to learn. So that message to all the students, that learning should be your real motivation. So what you're there for. So that quality of Shubham really struck me. Then other things, as he said, he was not very fluent in English, that I observed when I asked him to do seminars. But what I observed in his seminars, that he has gone so much into depth, and he was so much clear about the concepts and the science behind it, that amazed me. And language, I don't think should be a barrier. As you can see, now he speaks very fluent English. So that language can be, I mean, acquired, you know, but science has to come right from your mind, from your heart. So that is what my second message is to the students. Don't think that language is an important thing. Language is secondary. The third thing I found in him is I asked him to write an assignment. I used to teach fungi. I used to teach virology. So in all these subjects, I found the assignment that he submitted was a voluminous one. So it would be almost like a book. You know, yes. So that kind of perseverance and that kind of dedication, I noticed in Shubham, and from that day I was sure that this boy will go a long way. The type of dedication that he has, it was tremendous. I mean, in very few students I see that patience, that you keep on sitting and writing so much. The people get bored very fast, you know. They just write a few sentences, a few pages, and then they're bored. They don't do that. But that kind of dedication, it was almost a voluminous book. When he gave me, I felt the weight of it. And the content I also read, word by word, from end to end. And that was really marvelous. So I really, that one quality also really amazed me. And again, reminded me that here is one student who's very bright and will go a long way in science. The other experience that I had, I attended a primary immunodeficiency symposium. It was an Indo-US symposium that was organized in Mumbai. So I attended that because I wanted to learn about this PID, because I keep hearing about this, and it was kind of a workshop. So there were a lot of stalwarts coming from the US, particularly from NIH and other places, and I happened to meet his guide. And I told him that Shubham is my student. Then he said, he's amazing. He's such a nice person. So even talking to him, I could understand that his guide also, his mentor also was a very nice person. So that was another thing. Then I met Dr. Sujit Singh also. He is such a dedicated person, and Shubham has worked with him, so I could understand that two dedicated persons when meet, there has to be some miracle. And so that was it. So I told Sujit Singh also that Shubham is my student, and he was very much happy to know that. Then other thing, I went to PGI for immunology conference, and there I met Dr. Sakya, who did with whom Dr. Shubham did his PhD. Dr. Sakya was such a down-to-earth person, such a humble person. He talked to me about Shubham, and I talked to him about Shubham. I almost narrated all this, what I have told you. And he said, you're very right. To get such a student is really a blessing. And Dr. Sakya told me that he's a different kind of a person, you know. Dr. Sakya is a lot into music. He has his own songs, you know, and he showed me his album. So that kind of nature also is, I mean, I could reinforce when Shubham said that it's very important that what kind of mentor you get. So I can understand that. So to get good mentors to a good student is really a blessing for both. So with that, I thank you, Shubham, once again. I went to NIH once, and there I could not meet you because you were at that time in India. I really missed that opportunity because you would have shown me the labs and other things. So with that, I just want to say all the very best, Shubham, to you. God bless you. Thank you.
Thank you, sir. Sir, I still have that book for you. It's still there in my PhD lab. My juniors were telling me that your book is there. And I said, it belongs to Dr. Srivastav.
No, that was almost a thesis, you know.
Thank you, sir. Thank you so much.
Thank you. Thank you, Dr. Srivastav. And so lots and lots of memories are there with him, with Shubham. And so, but we have to value the time. And if there are any questions, any specific questions, or I would like to ask a general thing which will benefit everybody, that I will do in the end. Are there any questions, Bharanjit and Nupur?
No, ma'am. I guess everyone is so mesmerized by his journey.
Yeah. So I think everything is clear. We really enjoyed your journey, and from very modest origin, you were having that grit and determination, of which we are the witness, and we are so glad. Yeah, please, Dr. Gopal Krishna, please ask your question.
Thank you, ma'am. Good evening. I'm not a doctor. Shubham Goel, sir, story, journey was very inspiring. We are inspired by medical microbiology, and I am a student of BSc Industrial Microbiology. So, I mean, can I switch my stream from Industrial Microbiology to Medical Microbiology? Is there any provision of that kind?
You are right now in
Thank you.
BSc Industrial Microbiology.
Yeah, yes, sir.
Yeah, there is a chance that you can go. It's just that you need to give the entrance exam. And if you want to go, the medical microbiology is either you can go for the South Campus, though it's there, but I'm not sure there's a medical microbiology or not, but maybe a few labs there, but at least there's a microbiology. It's more of plant microbiology over there, I think so, in the South Campus. But in the PGI Chandigarh, you can go for the medical biotechnology, and you can go for the biochemistry. That is a medical biochemistry. But for medical microbiology, there should be some institute. I'm not aware right now, as of now.
No, he should prepare accordingly. Institutes are come later. How he should prepare? What he should study? Tell. The basic things like biochemistry, molecular biology, all those things he should study for the entrance exams.
Yeah, that's the one thing exactly. If you want to continue, because at master's level, actually more or less, if you go in the biotechnology or the microbiology, it's more or less same study. The course wise is the same, right? So for that, you need to give the entrance, and that to clear entrance exam, you need to read all molecular biology, cell biology, immunology. So all these subjects that we read in the microbiology in Delhi University, basically. So that you need to read all those.
So I want to ask something, which if you answer, it will guide all the younger generations. If anybody wants to go abroad, then yes, what preparations and at what stages should they start making? Some tips, because sometimes it becomes late when a person wants to start preparing. So what was your strategy, and what all you followed and you want to tell, what all even you missed, you may tell them, because we want that they should be even better than you and me.
So there are actually, if you want, and I would definitely encourage that you should come abroad, because I mentioned you before that the work of culture is entirely different, and you will learn definitely more and more here. But now there are two ways to decide to go to abroad. Either you go for the PhD, but again, in the PhD, if you come here, then you have to clear some kind of language test there first of all that, and then you go for the each university's program, PhD program, and which is very expensive, which you can, I believe that it's not affordable. But to overcome that issue, if you want to join PhD, there you can do by getting the fellowships, which I mentioned there. Those are only for the PhD. If you are really interested in PhD, so just go for those fellowship. And what actually in those fellowship, they are mostly needed. They needed your actually aptitude towards the research. So you need to make the statement of research, like why you want to come here, why you want to join that particular department or the lab, and what is the future vision. So that's mostly basically they ask in that basically fellowship form. And once you qualify, everything is covered by those fellowship, and then you can join a PhD there and you can do it. So that's the one thing. If you do your PhD in abroad, The other way which I did actually, because I told you there is something which makes you actually difficulty in getting PhD, and because of its cost, because of the other issues. The best way I would say, if you really want to do, then come after PhD, do your PhD in India, and then come for the postdoc. So postdoc is actually nothing. It's basically a research experience program which you get after your PhD. In that way, you don't need to apply any kind of fellowship, nothing. What you do, whatever your interest, like for example, what if you're in the cancer or immunology, you look for a person who is doing already in abroad, and look for her profile or his profile, see if it is a match to your interest, and then apply them and tell them that I'm this so and so guy and I've done my PhD in these subjects, and you show your publications and tell your interest, that how it matches to his area of interest. And if it likes you, then he will take the interview and he will ask you question, maybe 1 or 2 rounds, and then you all set. Then everything will be covered by the abroad university or the institution. So you don't need to pay anything from your own pocket. So this is the second way. So although there's nothing wrong that you come for the PhD, but there's also chance that if you have done your PhD there, you can come for the your postdoc fellowships.
I want to add something. A few days back, I was attending a talk by MSI, some other state unit. There was Dr. Suhas, who did his PhD abroad and came back. So he was saying that when you want to go abroad, you should make a statement of purpose which has a problem that pertains to the world, something that the world needs, where there's a niche. So try to write your research statement about that and involve the latest techniques. And also mean it, not only just to show, but mean it also. So then your chances, like the group leaders, liking your SOP become much better. And of course, you need a letter of recommendation, and the people, the teachers with whom you have studied, they know you the best. So they will not write wrong things for you, because we want to write authentic things, because we have a reputation. So we cannot tell lies, and it's unethical also. So whatever you have done with your, at the stage of doing your BSc and MSc, whatever traits you have shown, they will be very useful. Because if we vouch for a person and the person goes there, then that person has to match to what our predictions are. So the statement of purpose and LOR, letter of recommendation. So how to select a research problem, that I want to add. And also there's an input. Yeah, there's an input. Akanksha Puri, she's also our coordinator of Delhi unit. She is writing. Jamia Hamdard has also medical microbiology. One more input. She has done from there, and she is an ace in medical microbiology. She's doing her research PhD at AIMS presently. Okay, so I think you have got a lot of motivation and also some know-how what matters. Like Shubham sir told all of you, that keep your personal life also very full of fun, recreation, because it balances you and makes you what you are, and helps you to take a lot of pressure, work pressure.
True.
Not only that, you enjoy your work if you are stable at the home front and in the personal life. So we saw sir's 2 cute kids and a very dedicated wife in the picture. Many congratulations and blessings to you, Shubham.
Thank you, ma'am. Ma'am, I want to say again, I'm really thankful that it was you for the first time who provided me that platform. And again, it is the first time that you provided me this platform to present myself.
Shubham, actually, we are honored by your presence. You are we have so many worthy ex-students who have made it big in industry, academia, research, teaching, everywhere. One is here, Dr. Abhishek Saini, who is at Medanta. So he's doing the HLA typing over there. And lots, we have countless precious pearls in our kitty. So God bless you, Shubham. And you are at the top, and one among these. And it was a successful session. And Professor Deshmukh also has praised
especially
Yeah.
thank to him.
I have a question, Shubham, if you permit me. Can I ask you one thing?
Yes, sir. Yes.
Yeah. So this Card9 deficiency,
Yes, sir.
as you said, it leads to infections by fungi, like
Yes, sir.
Candida, right?
Yes.
And then you have the STAT3 leading to the
Yes,
infection of Staphylococcus aureus. I
Yes.
read somewhere that DOC8 deficiency would present itself with viral infections.
True. That's true,
Can
sir.
you tell me, because I'm always interested in viruses, so can you tell me how exactly, what is the mechanism and why only viruses?
That's very interesting question, sir. So that's what I'm actually doing right now. So now that's why I just did the new lab. So that's what we are looking. You actually wonderfully observe this peculiar thing, that in one deficiency you just have the fungal infection.
Right.
In the SART3 you just have a bacterial as well as the fungal infection.
Right.
But where the third gene, if you look at the DOC8, you just have the viral infection. So just see the unique susceptibility.
Yeah,
so
that's
how
what I'm wondering. Why only virus?
Exactly. Right. So, sir, your question is right. So probably actually I'm doing this right now. So what I'm doing is that I have my DOC8 patient and START3 patients, and as well as I have the mice models for the START3. What I'm just taking the skin samples of the each, and I'm doing the single cell RNA sequencing. So I would like to mention to those what single cell RNA sequencing, where actually in the single cell you can identify all the transcriptome at the same time. So you don't need to go one by one, like isolate the one single, like every single type of cells. So in one sample, like in the skin cells, like you have a fibroblast, you have stromal cells, you have a the hair follicles. At the same time, you have a data of all together at one tube. So that's the beauty of this single cell RNA sequencing. So that's what, sir, we are exploring right now. So we are trying to explore like how a single gene susceptibility is different or unique. So I cannot tell you what is the mechanism and how it is going on, but we are looking as of now, right now in my new lab.
So you're working on that mechanism, you're trying to work out on that.
What I'm doing right now.
And your approach is single cell RNA-seq.
Yes, sir. Single cell RNA sequencing.
Very good.
So probably if I got new data, I would publish or I'll show you.
Yeah, please share with me. Huh? Thank you so much.
Thank you, sir.
All the best for that. And Professor Deshmukh, sir, do I have your permission to proceed for the vote of thanks now?
You can conclude now.
Okay. Thank you so much. It was very enlightening. And I now invite your junior from the college, Dr. Abhishek Saini, who is with us from Medanta and belonging to the Sukshma family only. So I request Abhishek to propose a vote of thanks on behalf of MSI.
Good evening, one and all. First of all, I would like to thank Honorable Professor E.M. Deshmukh, sir, President of MSI, Dr. Jyotsna, who brought all of us together on this platform to share our views and work for microbiology subject. On behalf of MSI Delhi team, I would like to extend my heartiest thanks to our speaker, Dr. Shubham Goel, our Shubham Bhaiya. Thank you, sir, for enlightening us with your inspiring, motivating words. You are an exemplary person, and we have a lot to learn from you. Sir, with your research, you have answered most of the question without asking. So every student here, let it be a graduate or postgraduate, everyone is benefited from this lecture. And not only these studies, but the other, as you mentioned, Card9, TCF3, these deficiency, and sir has finally, Dr. Sunil Srivastava sir has finally summarized the whole thing, what you were trying to tell us. So it was in a very simple way and very easy to understand. And thank you, sir. If I'm not wrong, your parents are also a part of this meeting. Namaskar, sir. Namaskar, ma'am. Thank you for providing us such a wonderful senior. All the scientists, teachers, especially Dr. Sunil Srivastava, Dr. Parvinder, Dr. Lakshana, Dr. Shweta, thank you for blessing each and every team member, and heartiest congratulations to Dr. A. Archana, President of Delhi Unit, and Coordinator Dr. Shweta, Dr. Ms. Akansha, Thank you all. And here we come to an end of today's webinar. And the good news is that recording will be uploaded on YouTube for your future reference. Thank you. Good night. Have a great time ahead.
Thank you, Abhishek. Thank you so much for your great work.
Thank you. Shubham, are your parents also there?
Yes, ma'am. They are looking at me as well.
Okay. आपके parents को बहुत नमस्कार. Unmute करके बोल सकते हैं तो बोलिए एक second, please.
Yes, they must be proud of Shubham.
Yes, and we thank them for giving us their Shubham. Thank you.
Okay.
Thank you.
Thank you, Shubham.
Thank you, everybody.
Thank you. God bless. Thank you.
Jai Hind, Jai Bharat.
Jai Hind, Jai Bharat.
Jai Kisan.
Jai Kisan.
Jai Jawan.
Jai Jawan, Jai Kisan. Good. Very good.
Awesome. Thank you.
Very good. Shubham, keep it up.
Thank you, sir. Thank you.